Clone Wars. There's wars happening everywhere, and this is a different kind of one. We're decomposing aggressively tonight. So we're here tonight with Art, who's in Austin, and Dwayne, who's down in San Antonio with the crew at the Meme Lab. And I'm going to do a quick little intro and talk about why we are doing Clone Wars and some of the cool projects that we have going. So yeah. So welcome everyone. Give us a thumbs up on the chat. Let us know if everything looks good. And my name is Angel. I am the director of the Central Texas Mycological Society, and I do all kinds of fun things. And we'll talk about some of the other upcoming SporeCorps events. We had a really fun event this last Sunday, Austin Davenport. At our HQ in Austin, we learned how to do a still air box, and we also learned how to make a monotub using low-cost methods, and everyone went home with their own sterile spawn, like a master's mix or a CVG mix. I think I said that right. But yeah, so we're doing Clone Wars as an online way for us all to gather. We're doing it monthly, the second Tuesday. So we want to keep it going, maybe even through the summer. We'll see how things go. But visit the Clone or the SporeCorps page if you want to sign up to stay connected with everything we have going on. And if you have your own kind of lab set up and you want to be featured, you have some cool projects you want to talk about online, definitely let us know what you have going. But yeah, this community is here to bring everyone together, help us learn sterile techniques. And so tonight our focus is going to be on turkey tail, but we want to hear how we're doing. So I just ordered new t-shirts. They're getting printed right now. So let me play this again. The shirt went away. So we have a new shirt coming out. Whoop! Here's a QR code to get 10% off. But they'll ship late this month. So in the next couple weeks. And tonight, our fighters in the labs in Austin, we have Things Mycological, Art. And Art, here's some of his stats. Art, do you want to talk about some of your lab contamination, some stories? You have some stories to tell? He might have stepped away for a second. And then from San, you're there? Yeah, give us some details on what you have going down in your lab, or some past stories. What are some of your success and horror stories?
I think he's muted.
Oh, he's muted. Art, unmute.
Does this work?
There you go.
How about that? There's like a million stupid adjustments I got to make on that Zoom. Hey, look at this, contam. Oh, look at all that gross stuff. It's beautiful, isn't it? Look at those colors. You don't get those colors except with bacteria. Mustard yellow, coral red. Yeah, that's just part of the deal. And I like to work with spores, so I get a lot of contamination. And that's just part of the game. And what you're going to see today is going to be some spores that I've left for a little too long.
And we'll get started with the demo after we get done with the
Mm-hmm.
slides. But this is just kind of giving maybe some past history.
Oh yeah.
Yeah, and then like, what's
Yeah.
your
So
favorite?
I got into this when I was younger. Like most people, um, start tripping over mushrooms in the field, and next thing you know, you're running a mail order business, selling spores. And then, years later, get back into it, and I'm really into the sort of applied side of it. So that's what we're kind of working on here. But I also just like every aspect of it, the lab work, the field work, foraging, identifying, all of that. I got to ramp up my taxonomy, but yeah. And I'm also a creator of this little thing called Myconotes that is going to the printer this week. I'm very psyched. So keep your eyes peeled for that when it comes out.
Nice.
And we're gonna
Yes.
take apart this guy.
Yeah. So Dwayne, you want to give us some of your give us a little overview. Tell us what makes you fascinated by fungi, wanting to do sterile work.
Gosh, what makes me fascinated? Gosh, everything about it. I mean, just the way it grows, the different morphologies that come out, the challenges that arise, um, the 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 challenge of how many cultures I can keep at one time. They're never ending. So I don't have any cool contams right now to show, but I've had my fair share. Here recently, I discovered that even when you're getting over the flu and you're wearing a mask, coughing in the general area of plate work doesn't work out too well. Even when you turn away, you get all kinds of things growing around the edges. So it's learning something new.
Nice. Well, as I mentioned earlier, we're working on cloning. It's turkey season. It's turkey hunting season. Art, you said you found some out in the wild recently?
Yeah, Sunday I came back with a bounty. Unfortunately, got very unhumid on a couple of those days. So I think a lot of these, you can see how this is curling up. That was a piece of bark, but this is one of these kind of toothy crusts. All of these are growing on ligustrum, and some of them are familiar to me, and some of them are not familiar to me.
Yeah.
So the idea is to find and catalog as many of these, and to kind of test and see which ones are the hungriest, and break
Mhm.
down the slash faster. And right now,
So I
I
think
have.
the ones that so turkey tail, as some of you all know, it's also considered a medicinal mushroom. And here's a good kind of photo of like what false turkey tail looks like on the underside. It's like very smooth, paper-like. It's a lot thinner as well, but you want to look for this like pore surface that's visible to the human eye. And we are using it to cultivate and grow for removing ligustrum or helping decompose ligustrum in Austin's urban forests. And so ligustrum is a very highly damaging invasive plant that gets removed by different groups, um, different conservation groups. And gosh, like within, before we were even a mycological society, there were mycologists here in town that would keep different strains that were found growing on ligustrum, keep that alive and like keep grain spawn going so that when log inoculation season is here, folks can use it to decompose the sort of fuel or the logs a little bit faster. And so in 2019, there was a study done here in Austin and published in Cambridge with Jim Stack and Lisa O'Donnell from the city of Austin. And the study found that inoculating the ligustrum or the privet logs with turkey's tail accelerated decomposition. And it showed that it decomposed the logs about 11 times faster. So transforming something that might have to be like hauled off, hauled away, using more fossil fuels, could just remain in the forest and get the nutrients could get cycled and be available for the plants there. And so ligustrum sort of takes out the habitat of other native species. So I don't think I mentioned that earlier. So that's the reason why it gets removed by a lot of people that are doing restoration work. And so I just threw in here a little bit of like the health benefits in case you're curious. So, not only can it help decompose the logs, you can actually forage the turkey tail and use it for your medicinal. Like people have used it in like soup stocks. You can use it in teas. Some people make it into powders and use it for different health reasons. So, like I mentioned earlier, we want to see you in person as well. So we have another workshop coming up in April on the 11th. Hector will be doing some fun, sort of like kitchen witchery. He said he's going to get Rachel Ray and do some pretty cool stuff with like organic brown rice, like showing you all how to use simple things like coffee cups to cultivate mushrooms, like on a very low budget. So so we have a few more tickets left for that one. And if you want to sign up to just be first to be notified about events, scan this QR code. And we also want to, if you want to be featured, you want your lab to be featured, you want to show us your capable hands doing work in the lab. We are always looking for more people that have home labs that want to share some projects. And if you have an interesting mycology initiative that is aligns with our goals as an organization, we'd love to feature you. So let's get started. I'm going to stop sharing my screen and then we're going to pin Austin. And so everybody can see what he's got working. So 1 second here, I'm stop sharing. All right. So let's get started. What do you have going, Art?
Let me try and just zoom in just a hair without ruining the perspective. That good? I think that's good. So yeah, what I've got is going here, as I mentioned before, is a couple of turkey tail species, specimens, I should say, that I collected. Then these are all from spores and various stages. And as you can see, this spore print was taken exactly a year ago. How about that? All right. And I inoculated these plates back on February 12th. So it's a little long to leave them this way, but I wanted to make sure I had stuff for this demo. And then I also have that blue maitake that you had given me, that block. So I was able to get something off of that mold covered mess. And yeah, like I said, I love working with spores. I collect them. I make sure I label them, date them. And what I do is I give them a letter, and that's how I keep track of which one they came from. And then if you're not familiar, with all the little things on here, everybody kind of makes up their own, but TV, Trinity's Versicolor, T3 is the third transfer I made from the original, and this came from a liquid culture biopsy. And you're like, what's a liquid culture biopsy? Well, how could you biopsy a liquid culture? I'll show you. When you leave your liquid cultures too long and let them get all sad and lonely, and they start doing weird things like growing on the surface and trying to get out, like this poor thing here. I actually got part of whatever that was trying to become a fruiting body and got it on here just so I could get some of that mycelium. And it looks like we got some really good stuff going. So we're going to go ahead and just kind of get started there. I like to just show you what I work with. I try and keep it as simple as possible. When I'm doing tissue transfers, I'll prefer to use tweezers rather than trying to cut a section out with a scalpel. I just find it easier to just reach in, grab a piece, and dunk it right into the agar. So I have a couple of different tweezers that I use for that. Just a torch for sterilizing, sanitizing, and lots of 70% ISO. Go through a lot of that. And then I also use Everclear, 95% ethanol occasionally when I have some contam issues on something that I want to clone. The Petri dishes I pour myself. You'll see some weird affectations in there, grains and stuff. That's some caramelized something. I like to experiment with different recipes of agar. There's no one recipe, but these are malt extract, in case you're curious. And yeah, let's go ahead and get started. I'm gonna put a glove on. What am I doing? Glove. And I've already wiped this down with ISO, and I'm about to turn on the flow hood. So it's going to get super loud in here, and hopefully you'll still be able to hear me. Okay. All right, how's that? I'm going to have to talk loud. Everybody hear me?
That's perfect.
Let me just go ahead and start doing this. And I like to just coat everything that I'm going to use with a healthy dose of isopropyl alcohol. I don't do it all at once. I just do them as I take them. The other thing I wanted to say is I don't use parafilm. It's way too expensive, and it doesn't really do much different than grafting tape, which this is. It's cheap, very flexible, and you can do anything you want, and it sticks to these things just fine. Oops. Oh, that's not a good advertisement. But I promise you they work. So I just kind of, spray everything that's going to be in contact with, cleanliness that needs to stay clean. These I'll actually flame sterilize, but I like to just make sure that they're good and covered with alcohol just to get all of the incidental stuff under control. And then generally speaking, you work clean to dirty. So keep the clean stuff in the front and the dirty stuff in the back. So if you're going to do a transfer, you want your clean dish in the front and your dirty dish, your potentially dirty dish in the back. That way the flow of air is just going to take everything away. Also, do things in order of complexity and dirtiness. So I've got a specimen, live, natural specimen here that I'm going to try and get something out of. That's the last thing I'm going to touch, even though I just touched it first. Sorry about that. So let's go ahead and do a transfer of this biopsy from the liquid culture. I actually inoculated a couple of bags of sawdust that I made here with this one's cousin, and it's going wild. So I have a feeling this is going to do really well once I finally get it stabilized. Also, whenever I sterilize something, I'll keep it in the back here and just out of the way. This generally, I keep a rack about 2 inches off the bottom because I like to throw stuff underneath there that I'm not using at the time, but I want to keep it clean in the front of the moving there. So I'm going to take a clean plate and I'm going to spray it down. You could do it right here. The flow that's blowing all the alcohol onto me. And then just give it a nice little, rub down. This is kind of thin agar, but I think it'll still be good. The other thing is we're all right on these things with Sharpies. And black Sharpie generally holds up pretty good with 70%. Anything higher is going to just clean that off. So I tend to, things that I use frequently, I tend to use a little label maker and just do it that way. So I don't have to think about it. So here we go. We're going to just go ahead. Let me remove the film from that. Hit my hand again because I picked up that potentially dirty jar. And apologize for the ring light glare. That's part of the deal. Yeah, I like to make sure this is clean. You'll see here that this is not a brand new scalpel. I don't know if you can see that. This blade's been used quite a bit and sterilized quite a bit. All I do is cut agar. I don't cut anything else. This thing is razor sharp. There's no need to keep changing your blades if you don't need to. Anyway, I'm going to get over here and kind of turn sideways. And so you can see this thing. Hopefully, I can get it in the picture where you can see it turning colors. Get underneath it. But you want this thing to get red hot. And I like to take it all the way up the blade and just kind of keep the torch going up the blade and heating up this whole section here. It's just got a lot of nooks and crannies that anything can kind of get down into. But we're not going to use a whole lot. Again, we're just going to use this front part of the blade, cut a little section off, drop it in, and we're done. So here, let's go ahead and find a nice dense section here that looks like it wants to become fruiting body. Oh, there's a bunch of liquid condensation on that one. So I'm going to take a little, see how it's turkey tail is going this kind of thick and chewy. So I'm just going to like grab a messy piece here, that one. And you don't need much, very little of this to, in fact, here, let me get this other section so you can see that better. I'm done with this plate after I do these transfers. So I'm not being too careful with it, but I just want to keep up appearances here. So here I'm going to keep this open as little as possible. Just going to dab it right in the middle. And it wants to stick to the blade, of course. That works. That always happens. Exactly what you don't want to do. Look at that. Oh my God. All right, there you go. We got it. Look at that. Mycelium. All right. So then when I'm done doing a transfer and generally doing a lot of these at once, I'll just kind of put them under here in order of how I did them. And I'll go back and tape these afterwards, but I will forget which one is which super quickly. So since I put this in one dish, I want to sterilize it again, just in case there's some contamination there. I don't want to bring that over into the new plate. Always reapply in between everything, because everything you touch has potentially got something on it. I'll do the scalpel last. So let me hit this with another spore. This one is going to be fun because this is the random genetics. What you get with your liquid culture and your fruiting bodies, clones, is a clone. That's why we call it a clone. What you get with spores is potentially completely random genetics. So these spores contain all the genetic information as the fruiting body that I picked, but they're going to express themselves in a completely different manner, no way of knowing. So let's go ahead. I put that in the front, I put it in the back. You get another plate. You spray that one. This one's especially speckled. It could be the, I think I put nutritional yeast in this just to see how that would work. Apparently works pretty good. All right, let me go ahead and make sure this is clean without any kind of debris. And then I'll sterilize it again. If you can see what I'm doing, let me give it here. This is just a butane torch. Anything that can get it hot enough. A regular lighter is really not going to get it as hot as you need it to be. It might make the very tip glow, but not the rest of it. So let me just get enough of that. It'll feel comfortable and get in there. And we are going to take, so this is the plate I dropped. So you see this little thing down there, that's where the agar cracked before I inoculated. So some stuff got down in there and got really familiar with itself. So I'm kind of hoping that that's going to give it a little extra oomph when I transfer it. So I'm going to cut right across that and get a piece that's got both hemispheres, both sides of that gulf, and stick it in there, just like that. And then hopefully you stick just like that. All right. Let me see. I know we're kind of running out of time, but I wanted to try and maybe do one of these. This is a mustard yellow polypore, and it's very tough, like hard, like wood practically. I can split it with my hands and get to the inside of it. That's what I'm going to try and do, which means it's kind of a multi-glove process. So the first thing I want to do when you're working on anything like this wild is you want to spray the living hell out of it, every bit of it, okay? You're not going to eat this, and don't worry about hurting anything. That's exactly what you want to do. So I just kind of spray them all down really good. And then make sure that the part that you're wanting to manipulate, let's say I'm going to try and split it right down the middle here in two parts and get to you can see the bright yellow part, that's the living part. This is kind of the dead part. So I'm going to really kind of douse it here. There's no guarantee that this will keep the contamination out. Like I showed you before, can plates, it's real. And these were spore transferred. And this is what I got. But so were these. So it's a numbers game. Just do enough of it, and it'll be okay. So what I need to do first, which I actually forgot to do, I'll do right now, is really get that. You can't see what I'm doing, maybe, but I forgot to spray this down and remove the film so that it can do this in as few steps as possible. I have another piece of film on there. Get it off of there. Fingernails when you're wearing gloves. You see, it works too good. Oh, here we go. Okay. So let me just get that out and be ready to go. Right here. Douse my hands again. We're going in. Sterilize my scalpel and have it at the ready. Here, I'm actually going to turn the gas up a little bit. We're really going to get this one. Oh, sorry, not in the camera. This whole multi-camera thing. We're two is live. Now we're getting things a little blow now. I'm still assembling all the parts for my induction sterilizer. I need one more part. Ridiculous. Okay, so this is ready to go. I can leave it here. This whole thing was, doused with alcohol. Nothing is going to touch the blade. I can leave it here. I can have this thing at the ready, open. I'm going to grab my weird little specimen here. I'm going to try and split it this way. I'm not going to use a knife or a scalpel because I'll end up stabbing myself. This is just really hard. stuff. So I'm going to just try and break it here. I ended up breaking it a little too close. You can see the gills in there, or rather the pores, rather. But I want to get a little deeper into that. So I'm going to try and get something down in here. And I can kind of see maybe a little more orangey down in there. I also see some other things. There's some mold right there. So I want to be really careful, and I'm just going to grab a piece of this and realize now that I would rather use my my tweezers for this. So I'm going to switch over to the tweezer, and let me just get a little alcohol. This one, I believe I already flamed for this one. So I'm just going to grab, let's see if I can get up here close where you can see it and get it in focus. Hard to get this to take in focus. So I'm going to try and grab something from in here. And I can already feel the tweezers kind of getting in there. It feels a little soft. You don't need much. Mycelium is 1 micron. So here is, whoa, I'm going the wrong way. You see how much I have here? I'm not in front of clean air, so this will probably be even more contaminated than I am out. But I'm going to grab that now. I'm just going to tap that, and that's it. I would normally do more of these, and I'll put 3 of them per plate because it happens really fast. This one I'll just do the 1, but you know, imagine me having done 3. And that's it really, as far as like the cloning goes. You're really just grabbing a piece of tissue from the inside because that's as close to being, you know, bug free and mold and other spore free. Yeah. Anybody have any questions? I'm just kind of flying through this, and I'm realizing I don't have the chat up or anything, so I can't see if anybody is actually even here. I can go on. Anybody there?
Hello, hello. We're here. We're here. So I removed the pen.
Get the tape going. Say hi, tape these up.
So there were a few little comments that came up.
Yes.
More just like tips in the chat about when to label your agar plates.
Mm-hmm.
Is it better to do it like before you start doing transfers or and like spraying alcohol, or like before you do your work, kind of like label them?
Oh, no, you're setting yourself up for disaster because you'll drop a plate. You'll, you know, try and get the piece of agar you're transferring to stick 10 times and it won't go. Yeah, I just wait until I'm got success. And then, like I said, I just kind of keep them here. I know exactly how to label both of these. And this is the only cloning specimen I did today, so I'm not going to confuse that. But yeah, I like to just kind of do it afterwards. But it's very much get a system so that you can keep track of things. I can't tell you how many times I forgot what was what.
Mm-hmm. Well, I guess we can see next month like what happens with that mustard polypore. Maybe we can make some hot dogs.
I'm gonna do some more transfers later on tonight and be a little more kind of strategic. I just wanted to show you guys the basic premise there. Oh, and the other thing is I do use colored Sharpies as well, but they will completely wash away with 70%. So you can get away with, you know, the black Sharpies when you're kind of spraying, because I mean that's what all these were, and they're still readable, but you know they'll fade a little bit. But any of the colored Sharpies seem to just run immediately, and I found that out the hard way when I was like spraying everything down and suddenly I didn't know what anything was.
I'm going to show your face now. So, do you want to kind of like share some things about kind of like what you've kind of encountered, like looking for different fungi growing on ligustrum, and maybe share a little bit more about kind of like, what you know about it as like an invasive species and why we want to.
Yeah, it's amazing. I don't know when it was introduced here in Texas, but it was very popular in the 1950s, especially the Chinese ligustrum, one with the little leaves and millions of shoots going up. But it, quickly escaped into the creeks, and all the runoff goes to creeks, and the creeks go to the river, lake, sorry, and it just spreads. The birds, really, the cedar waxwings are the ones I see most prevalent, and their gullets are just full of berries. And what do you call it? I don't know what's happening. Anyway, the they get drunk on them. Apparently, something ferments in there. I don't know. That's what I've read. And they'll fly into your window, and we've got big windows that have purple bursts. It looks like somebody threw a water balloon full of grape juice, against our window with a bird splatter pattern. But it's really insidious in that it destroys the understory, or I should say, it changes the understory in a way that native plant species, insect populations, and other sort of fauna, soil fauna, and everything. It disrupts the cycle because the leaves are more rapidly decomposing, and so they go through the cycle faster, and that screws up the equilibrium of everything else around it. The other thing is it's mostly evergreen, and it can produce berries 3 times a year. So it's really just all out. There's no controlling it. But there is, like we're working on here, you can cut it back. But what do you do when you're deep in the Belcones Canyon Land Preserve, where you can't drag these trees out? You inoculate them, and hopefully you can identify a local strain, and you can get them in a bag, like this ugly bag that got all weird in my pressure canner. But this is one of those crazy looking liquid cultures that I collected. And here you can see some bands of it going through there, going all over the top. When I inoculate these, I just get the regular bags with the patch on it. Some people just get a bag and buy some of these pouches. Some people just use Ziploc bags. And then I bought this sheet of these little silicone sticky thing that sticks onto the bag, and it's kind of a self-healing injection port. You're never going to use it again. You're never going to inject through there again. So it's kind of overkill. You can literally just inject through the plastic bag and take it off, and you're fine. But anyway, yeah, this is a 5-pound bag. 10-pound bags are kind of overkill for this when you're just trying to get a feel for how fast it's growing. But yeah, just for those bags, I just inject them with about 10 cc's, which is this, but 10 cc's. That's only 9. But this is actually you can see it, it's backwards, but that is the exact liquid culture that I took the biopsy from. So you can see it's very clear liquid, and the mycelium's all clumped together. That's a good sign.
Mm-hmm.
So this is what I'll use to inoculate a couple more bags. Just I don't want my reputation to be tarnished. I want to show some nice clean bags. Anyway, yeah, great order. I'll roll with my garbage.
Nice.
Yeah, and to date, I think I identified like 10 or 11. No, sorry, like 8 or 9 different species growing on ligustrum at various
Wow.
stages. And there's one that eluded me so far, but it grows on the roots. So I'm excited
Whoa.
about that because we've yet to identify a parasite
Yes.
pathogen that will actually kill it without killing all the other trees. So
Yeah, and that's something
baby
we're
step.
hoping maybe with like DNA we can find parasitic
Yeah.
species of fungi that already exist and grow on ligustrum. But if anybody that
And this is a problem everywhere. It's an invasive everywhere. So we can take that same data from other states and other regions who have identified a species of fungi that we might already have here, and start to cultivate that. That's
girdle it with fungi.
yeah.
There was a question that came up whenever you were doing the spraying down the mustard polypore with isopropyl alcohol. Someone was asking, can you use 3% peroxide? And they had seen this text somewhere else, and I've heard that same thing.
Yeah, peroxide. Uh, I don't think it does the same thing. And there's people, let's say on Reddit, who will argue, will die on that hill. But I just find that alcohol, and if you don't want to use isopropyl, like I said, you can use ethanol. But you could conceivably spray down the outside of a king oyster mushroom with this, ethanol, and when you're done cloning it, just let that dry. It'll evaporate completely, so it doesn't leave anything behind. So theoretically, you can still eat that mushroom, but I'm not going to eat this mustard polypore, no matter how delicious mustard sounds to me. But yeah, I would stick with alcohol to get
Yeah.
guaranteed results. And also 70% when you're sterilizing your gear, because it hangs around longer. It's the same COVID, idea. Like, don't use 99%. It just evaporates with immediately.
Yeah, we had someone joining from Western Pennsylvania, and they said ligustrum is also grows and is invasive where they are because people love it for hedges. I had to
Yeah,
pull some
it
out of
makes
my
great
yard
hedges.
this week.
That's
Just
the
this week.
problem. It's for sale at Home Depot. Nandina, for sale at Home Depot.
Boo.
Everywhere. Yeah, that's not going to stop. And people are
Yeah.
going to keep planting that. Tree of Heaven, whatever. There's commerce involved.
Mhm.
All of our feelings aren't going to make that go in. So you should, just kind of flip the script and figure out what we can do on the other end to help out our local species.
Well, Art, thank you for showing us that demo. Excited to see what continues to happen with the turkey tail.
Absolutely. I wish I could show you a better close-up of the Contam, because that, if you could see that
Oh, wow. Yeah.
color is, it's not coming across, but that coral color, I wish I could just capture that.
Yeah.
The other
That's
ones are like
pretty.
mustard yellow. Yeah,
You know what that is?
I have no idea. But it's sort of it. That's as much as they spread, and then they stop. They must not really like this agar recipe. If I had like more of a bacterial recipe, I'm sure they would go nuts.
So
But yeah,
do you I see sometimes people putting molds and stuff on iNaturalist. Do you know if like it's getting better, like pattern recognition is getting better with molds or
it's tough because you have to get so macro with molds to really see the differences. A lot of mold looks like mycelium, and just looking at it from a plate, like, oh look, I've got all this great mycelium. and then you come back the next day and there's all these black things.
Mhm.
It's yeah, I've yet to sort of find a good visual resource for that. ContamFam, r/ContamFam on Reddit is pretty
Pretty
fun.
good.
Yeah, that's a good one.
Mm-hmm. All right, well, let's move the spotlight over to Dwayne down in San Antonio. What you got going over there, Dwayne?
Mute.
You gotta unmute. Oop. Let's see, maybe some technical difficulties. Let's see what we got a little glitch going. What is a live stream without a few glitches? Hi, we can't hear you. Where's your audio? Is it being stupid? Huh. Are you able to join the same Zoom with your phone maybe? We could use your phone audio. What's happening? Oh, I see you're unmuted. Dang it. Boo. We can't hear your sound. Did you hear? I'm going to text him. Can you join via your another device to get audio? Question mark. Oh, I heard a blurb.
Can you hear me?
Yeah, there you are. What?
Hey, I
Wow.
had no idea what was going on. It was
Glitched out.
so sorry.
We don't even have like Mercury in retrograde or planetary alignments happening.
Yeah. Oh, sorry about that.
Wow.
My earphones got bored and disconnected or something.
Yeah. Well, we hear you now.
Well, I love what Art was saying. I agree with everything. I think we're going to get along just great. I didn't know. I didn't know so much about ligustrum, and I didn't know it was still being sold at Home Depot and the big stores. Is it still marketed under the same name, ligustrum or glossy privet or whatever it's called? Or do they get sneaky and change up the names?
Glossy privet.
Glossy privet. Wow, that's terrible.
I mean, if you Google glossy privet and
Yeah.
glossy privet pathogens, fungi that attack glossy privet, you're only going to get gardener and arborist sites for helping
Right.
you keep them healthy.
Right. Well, I got pretty much the same thing. I've got the turkey tail from Austin here that has taken over the plate. These were done on February the 10th. And this one's growing up the sides and up the come to the top of the plate there. So I can make a transfer off of these. You want to see that? Or I've got this Ganoderma here that was in a in a tube for 2 years. This was April 24th, 24. So whichever you guys want to see.
Ooh,
Either
I
one.
could maybe yeah, I would maybe so have you already put your turkey tail, like have you added that already to grain?
It is, yeah. It's already on the hardwood fruiting block growing out. It's about halfway done.
Oh, nice.
Yeah.
Yeah, so can you tell us real quick like what we plan to do with that?
So yeah, I'm gonna send that to the group there in Austin, so they can use it as a plug spawn for the same reason. They're gonna um, plug ligustrum logs that have been cut down to speed up the decomposition and get them out of there quicker, so that the dry weather doesn't spark a fire and light everything up. So I'm gonna get that going and get some more liquid culture going. I think the more we get going and the more sources we have, the better. The more diversity, the better. So
Mm-hmm.
what I'm doing with this.
Cool. I
Also
think
got
seeing maybe like is there any way you could do both and then we can see next month like where the progress?
sure we can do that.
Okay. So do you know what the Ganoderma was? It a sessile? Was it a wild one that was cloned?
Yeah, I think it was a sessile. It was a real funky, and the inside it was a real fluffy type of inner part of it, not like a like your standard multipilum or your typical medicinal one.
Mm-hmm. Yeah, I think that one. We want to start doing more stuff like because that one's what the species that's typically used for doing like leather and making like
Paper.
yeah, like and people are making like furniture even and things like planters, like planter pots, like candle holders. Um, but I've been told that that one like does pretty well, like and resists a lot of other contaminants, even in like our dry weather and stuff. So I've been wanting to do like a fun kind of create a mold and do a fun project and sculpt something um, with Ganoderma. But it's just a matter of like having stuff ready to kind of fruit out or to put on grain spawn or not on grain spawn, but I guess sawdust.
Yeah, yeah. So I heated up my blade, got that ready to go. As it gets stuck, it grows to the top there and it gets stuck.
Wow.
So I don't know if you can see that, but it's all grown up along there.
Wow.
So you can grab just about anywhere here.
I know in the class that Esley put together, there's the like different types of mycelium. Is that I can't remember the words. It's like
So, yeah,
tomatos and is that
tomatose
the
and
kind like the fluffy?
right. So this would be a tomatose type growth, and then the other is rhizomorphic, where you can see the actual hyphal strands reaching out.
Mm-hmm.
So if I have a plate like that sitting around here.
So now that you're done with now that you've taken a piece out and you've kind of expanded it, like does the mycelium just like kind of fill in the gaps, like where you cut some out and started another tray or another?
Here,
Yeah.
yeah, it'll grow in and fill in on the bottom. You
Mm.
can see a spot there. So in a few days, that'll probably be covered over with mycelium, just
So
because
have you
there's
ever?
still remnants there of the nutrient.
Mhm.
So
So have you ever had any species like eat through the plates or do anything weird or make their way out?
I haven't had them eat through the plates, but I have had oysters and the turkey tail here push its way out and start to grow out. The edges here either grow out or it'll start to lift the plate up and push its way up. Yeah, so it can get pretty tough and thick. Reishi can also do that.
Nice.
That's awesome.
Yeah, and I like to usually I'll keep even if I'm going to toss the plate that I transfer from. I like to keep it just so if I get contam here and I don't get contam here, or I get contam in both, it kind of helps me figure out, hey, where I messed up. Was it just a simple mistake? Was it already there? You know, something like that. So I'll
Hmm.
take it, just keep it for a while. And to the question of labeling and how you do it, I'm kind of like Art. If I'm doing multiples like I am now, I'll do one, set it on top, and then push it to the side and label it in a little while.
Hmm. Cool.
All right, now I'll do the panoderma. And I use the same stuff that Art does. I love this stuff.
Hmm.
Since I found that, don't touch parrot gum anymore. You can't go wrong with these torches. Although I am jealous of Art's induction sterilizer.
Don't be.
All
Hey, this is currently what it looks like.
Here, I can, you can put that back up. I'll highlight your, spotlight you.
There you go. That's what it currently looks like. A little 3D printed box. All this stuff. Just need one more part.
right. So it's pretty much the same process as going plate to plate. There's just a little more to it because you got a cap, you got to take the cap off. Got that out of the way. I'll try to do this so you can see it. This has been in storage for 2 years, but it still only grew about halfway down. So really slowed it down. But it looks nice and healthy inside there.
Hmm.
You put a popsicle stick in there, right?
I didn't in this batch, it doesn't look like, but I usually do unless I just forget.
And the way that it, so you, the, is there like a type of tray that you use so that it slants like that where you get the most surface area?
That's a little trick you got to figure out in your environment because the way I do them is, put this off the blade there, is I mix up my agar, then I pour them in, and then I put a bunch of them in a quart jar so they're all standing up, and then I pressure cook them that way. So when I take them out, I have to figure out how I can angle them and get them just right so that they cool at a nice angle like that.
Hmm.
Not always as easy as it seems, but once you get it, you got it, and you got a system, just like Art said.
Hmm.
So
And then, how long is that like the best way to store, like as far as like the shelf life?
I think typically you can. Eventually, it's going to run out. I also experimented a little bit with these little vials where you use distilled water. You
Uh-huh.
take a few agar slices and drop it in there. Um, I've had them up to 3 years. I still have some, a box of them stuck on the shelf over there.
Hmm.
I've heard people say it could be up to 25 years in that method. I don't know if it's been proven or not, but
Hmm.
seems seems to
I've
work
heard the
well.
same.
Cool.
Yeah. The thing with this is it'll eventually run out of nutrients, it'll,
Hmm.
but I mean, mycelium's tough. So even if it's, half dead and no nutrients, they
We
can
go
usually
through a,
survive.
we go
Yeah.
through a, we get our, the clock is pretty close to midnight right now. We have like another major extinction event.
Mhm.
That mycelium's going to be just fine in that slant.
Yeah.
So yeah, so lots of good comments from folks out on the live stream. And yeah, if people want to give us ideas on what you want to see next, we love to hear people's ideas. Like, is there any topics? I know there's a lot of complexity around just sterilization and, or not sterilization, like pressure cooking and pasteurization.
Sure.
So I'm curious if people want to do like a night where that's all we talk about is pasteurizing. And if,
I think that'd be a great topic.
Yeah.
yeah. What about, what are some other areas to let us know where you want to see topics, or even what type of, if you want to see some spore work? I know that's like getting into some advanced areas, but
And I
yeah.
would say that spore work is some of the lower hanging fruit, because there's no sort of expectation of sterility or in working with capturing spores. So it's,
Yeah, so maybe we do like
get
one
in
that's just like spore print, doing a spore print and cloning. And if there's any
yeah.
species too that people they're interested in, or yeah, or have questions about, let us know. But this has been fun. Another Clone War. We're getting on the top of the hour, so
I have a sticker.
appreciate everybody taking the time to join us and go deep into the lab. But yeah, I guess we'll see everybody next. It'll be April, what date? April the 8th. That's my sister's birthday. Boop. So yeah, we'll see everybody on April 8th for the next Clone Wars. All right, talk to y'all later.
Bye.
Bye, everybody.